Hey guys, so welcome to the last lecture of this semester. Um, I'd like to thank you for kind of hanging in there. I know this hasn't been an easy transition to the complete online forum for both you and me. So thanks for hanging in. Thanks for keeping up with the assignments, and thanks for watching the online lectures. And so now we're kind of gonna go from our bulk RNA sequencing activity or looking at microarray data to kind of what I consider the new frontier of RNA sequencing and NGS analysis, which is this concept of now being able to quantify gene expression in a single cell. And we're going to, the activity based on this, it's going to be extra credit. So I'm not going to, have any points assigned to it specifically. But if you do want more practice within R, this is a good opportunity and it'll give you a few extra points for the semester. So, you see down below, we've been talking about and we've been analyzing kind of bulk RNA sequencing. And so basically, we take, any particular tissue that we're studying, and we know, from our basic biology classes that a tissue is, a very heterogeneous group of cells. And so we see all of our different cell types in different colors here. Basically what we do in RNA sequencing, without getting too much into it, is basically take that tissue and blend it all up and then make it one RNA input sample into our NGS workflow. And so basically what we have at the end of the day is we have average gene expression from all of the different types of cells in our tissue. And so this makes it impossible for us to look at any type of cellular heterogeneity. So if we're interested in, what particular cell type, could be contributing to a disease, for example, we would have no way of telling, if we were doing bulk RNA sequencing. Whereas single cell RNA sequencing, we're able to tag these specific different types of cells and actually carry that special tag through the sequencing process, and it allows us to reveal specific heterogeneous types of cells and rare subpopulations within our tissue that could, be causing different disease or whatever our phenotype of interest is. And so we're not going to get hung up exactly on exactly, what goes on, in the experimental process. Just to say, that we're, the technology here, we're able to separate single cells, which you see here, the little dot, in what are called these GEMs or gel beads in emulsion. And basically we tag these with a specific barcode. And once we have our sequencing data back from our NGS sequencer, we can then use that specific barcode that's now part of our short sequencing read to trace it back to a cell of origin. And so in this way, we're able to get the gene expression profiles on a per cell basis. And so here's just an overview of the workflow. A lot of this has been done up front, and we're going to be at this particular step, data analysis. And so just keep in mind that we've already taken the cell suspension, we've made that library, and then we've sequenced that particular sample, and now we're here. And so here is kind of a very common deliverable of these single cell experiments. And so what we have here is what's called a t-SNE plot. And what I want you to understand about this is that each dot that we see here is indicative of one of those cells where we looked at the gene expression. And the closer they are together, basically tells us the more similar their transcriptomes are, the more similar the gene expression of those given cells are. And using computational methods, we're able to cluster these cells into specific clusters that might be informative later on. And so when we're kind of going through this, right now they're kind of arbitrarily numbered from 0 to 14, so we have 15 cell clusters. And now what we basically need to do is we need to look at specific gene markers that might be expressed within those particular cell subsets. And so, for example, we see CD3D, which is a commonly used marker of T cells, for example. And so we see our T cells light up as far as cells that are expressing that particular gene. And CD3E, as another example, we see nice overlap between the expression of those particular genes. And we can do similar things with other other hallmark genes that indicate what cell type they are. So you see here the expression of CD79A and 79B, which are commonly used as markers for B cells, if you know what those are within the immune cell profile. And we can see that this specific cluster at the top of our diagram here is primarily our B cell population. And using this kind of strategy, we're able to kind of annotate our cell populations and determine, what they actually are. And so you see here that we're able to identify our CD4 and CD8 T cells, our natural killer cells are down here, our B cells that we showed with high expression of CD79A and B, and then our monocytes, dendritic cells, all the cells that you've heard of if you've dealt with any type of immunology at all. And so I'm not going to dwell very much on this. I think, especially in this way, learning by doing is important. And so let's start our quick activity. Remember that this is all going to be extra credit, and so try and get it in pretty early next week, by Tuesday, hopefully, and it'll contribute some points to the overall course grade. But what we're going to do is we're going to look at a data set containing about 3,000 what are called PBMC cells, which are peripheral blood mononuclear cells. And basically, these are all of the nucleated cells if you extract a vial of your blood. So we're going to use a library in R, which is called Seurat. This was developed by Rahul Satija in his lab from the New York Genome Center, used quite extensively in the single cell literature, and it's a good program to learn. And basically we're going to use, these specific tools to do a number of things on this data set of 3,000 PBMC cells, including clustering our cells, identifying specific cell subtypes, and then finally do some differential expression comparison. And to get credit for the extra credit, I want you to remember, open a new R notebook, enter in your code, and then after you're done with the tutorial at the website above, I want you to then knit to HTML and then send me the HTML in an email. And if you have any questions, please don't hesitate to reach out. And thanks for listening, and good luck.